right, which of course was spasm of the smooth muscle and constriction of the airways. And in his book, he also had a drawing of sputum, and this was before Paul Ehrlich had developed the dyes for cells in histology. So those are just nuclei, and there are going to be eosinophils and neutrophils and so on in there. And you can see the Charcot-Leyden crystals, which is the eosinophil protein which characterizes the sputum of severe asthmatics. And he also mentioned, in terms of therapeutics, coffee as a potential treatment. So, you know, he was really quite a forward-thinking man at the time. But it really was Paul Ehrlich who sort of developed these aniline dyes, such as toluidine blue and eosin, that really enabled the cellular aspect of the asthmatic and allergic processes to start to be described. And I won't go through all the various descriptions of him here, but I think it's the differentiation of these cells, the eosinophil with its red granules and the mast cell with its metachromatic granules that characteristically describe the asthmatic inflammation in a much more precise way than had been done before. And we're going to hear about eosinophils and mast cells a little later on today, so that's great. The next step in all of this really came when William Osler, who was one of the founders of the Johns Hopkins Medical School in Baltimore, he came over to England as the professor of medicine at Oxford. And during that time, in 1892, he wrote his first textbook of medicine called The Principles and Practice of Medicine. And if you look up asthma in the 1892 version of that, you'll see these sort of descriptions here. And it really is truly remarkable, because what he did is he brought clinical medicine, physiology, and the beginnings of pathology with the various pathological stains that have been developed to describe an integrated view of bronchial asthma, including the very characteristic mucus plugs that were given special names, and also these crystals I talked about, you see on the right here, which have been described by a French physician, Charcot-Leyden crystals, earlier before he published his book. But basically, this man really started to put it together. That's what I would say about Osler, and of course he's a wonderful physician at the same time, particularly with children. He had a great way with children during his career. Now, moving on beyond eosinophils and mast cells, just describing them as inflammatory cells, the next big step was to understand how these cells were activated and brought into the inflammation. And this is Karl Prauschnitz, who lived in Germany when he was doing his experimental work with Heinz Küssner. And what they basically described here was a transferable agent in the serum that moved allergy from a person who was allergic to a person who was not. So he injected the serum, and in this case to cod allergen that Küssner was allergic to, into Prauschnitz's skin, and then challenged the area of skin with cod. He got this wheel and flare reaction, this characteristic allergic response. And so this was the so-called reagin that he talked about at the time. Well, in 1936, because of the uprising in Germany, and he noticed the politics was going wrong, he and his mother moved to England, firstly to Manchester, and then subsequently to the Isle of Wight, which is just across the water where I'm from in Southampton here. And he settled in this little village called Ventnor, which is a coastal village, and became a general practitioner there for the rest of his life. He was very much loved on the island, and they called him Father Giles, because his mother, who was English, was Giles. And when he moved to England, he changed his name from Prauschnitz to Giles. And he's left a lovely memory on that island about how he looked after patients at that time. So really quite interesting little history there. Well, what was reagin? Reagin turned out to be the fifth immunoglobulin class, and of course two groups were working on that in the 50s and 60s. Gunnar Johansson in Sweden and his group, and obviously Kimi and Terry Ishizaka working in the United States, initially at Baltimore, at Johns Hopkins. Initially at Denver, then at Johns Hopkins. And they define this reagent, this transferable serum agent, as IgE. And those are the two publications. And there's a picture there with Dennis Stanworth, who was one of the co-authors, as you can see on the paper, in Lausanne, when they registered the IgE molecule as the 5th immunoglobulin class. So this was real progress, because we had mast cells, eosinophils, we had a description of the physiology, and now we had the IgE system. The next bit in this story was, of course, the description of the chemicals that cause the asthmatic reaction. And Henry Dale really was the first person to describe histamine. Again, I won't go through all of this, but he was a pharmacologist who became head of the Medical Research Council and subsequently president of the Royal Society here in the UK, and got the Nobel Prize, as you can see there. But the important thing about Henry Dale is that he was a pharmacologist of the true pharmacology. So in a way, he's kind of the father of British pharmacology, in that he developed antihistamines to block histamine and a variety of other things. And he also was one of the people who worked on adrenaline in the early days to show its effect on the cardiovascular and respiratory system. But I'll come back to that later. So we were getting an integrated picture now that IgE, mast cells, eosinophils, chemical mediators like histamine were being released in the asthmatic lung. And people then started to focus in on how it all came together. And I put a few pictures up there of various people. I'm going to talk about K. Frank Austin in a few moments, but one of the people who really pioneered all of this was Jack Pepys, who came from South Africa in Cape Town to England in the 50s during the difficult time in South Africa there. And he was the one who really set up the early and late reaction allergen challenge work while he was working at the Brompton Hospital in London. And I've just drawn out the pictures that were in some of the books in the early 60s about what was observed, the early phase, minutes, the late phase, hours, and then this hyperreactivity of the airways that could go on for several weeks that Freddie Hargreaves and others worked on. Shown here is also Barry Kay, one of my good friends and colleagues who died fairly recently in London. He was at the Brompton Hospital, and he followed Jack Pepys into that chair looking at bronchial asthma. And again, you can see K. Frank Austin. So back in the 1980s, when I sort of started to come in on the scene, this was what it was beginning to look like. Early and late reactions, hyperreactive airways following allergen exposure, a whole variety of inflammatory cells, in particular, obviously the mast cell, the eosinophil, but now what we were beginning to see the lymphocyte starting to make its appearance, the T helper cell, as you can see on the far right there. And it was Barry Kay's group in particular who was very interested in that particular cell type, but also Mossman and colleagues in the US were starting to work on the type of T cells that were contributing to this eosinophil and mast cell and IgE reaction. And through the sort of early 80s and into the early 90s, we started to have these meetings, started off by Lawrence Lichtenstein and K. Frank Austin in the United States, but then Barry Kay and later myself and Gianni Moroni joined them, and we started having these meetings to bring together scientists from across the world who were interested in different aspects of asthma, the pharmacology, the physiology, the immunology, etc., etc. And here are several of them at the meeting we had in 1983 in Newnham Park in Oxford. And again, you can see some familiar faces there. You can see K. Frank Austin, Lawrence Lichtenstein on the front row. I'm looking rather young and innocent on the right there, but also if you look back through that crowd, there are some very famous people there who contributed to our understanding of the biology of asthma, including Tom Platts-Mills and Jane Adele and various other luminaries in the field at the time. So we had this, and what happened in the next 10 to 12 years was that those factors that were being described, eosinophil factors and mast cell factors and T cell factors, were being identified as real substances. And of course, this was the birth of the cytokines and the chemokines. And I've put them up there as some examples. So all of it was right, but we just didn't know what the molecules were at the time. And of course, now we do, because obviously these molecules have become targets for interventions, as I'll talk about in a second. So this was a very exciting time for asthma. We started to move from the phenomenology into some real immunology here, and it was a very, very exciting time, and people like Paul Byrne, in Canada were very instrumental in helping sort of populate this in a functional way, connecting the early, late reaction and hyperreactivity to various sets of these molecules. But then what happened over that period of time is that asthma was being recognized not just as an inflammatory condition, because obviously it could be a lifelong condition, and sometimes the airway constriction could not be reversed. And this really was the sort of birth of the remodeling, the idea that these inflammatory processes going on in the lung, in the airways, could actually alter the structure of the airway wall. And of course the epithelium and the underlying mesenchymal structures, including the smooth muscle, were an important part of this remodeling, and I'll come back to that a bit later on, but Emily is going to talk a bit more about it as she gets into her talk. So between 83 and 23, you'll see there's been quite a sort of set of discoveries that populated this disease with some real exciting new mechanistic insights. So when we now come onto the treatment, of course, it all started with Henry Dale, and he was very early in showing that antihistamines weren't particularly good in treating asthma. But even at that time, back in the beginning of the 1900s, they did have bronchodilators. There was this one, some of you will recognize, an anticholinergic agent that was smoked in cigarettes, which was quite remarkable at the time, considering you're treating a lung disease, but never mind. They did that, and this herb was grown industrially in India, and these cigarettes were exported around the world, and you can see some of them up at the top there. But that really was sort of first bronchodilator. There was ephedrine as well that had been developed in China, Ma Wang as it was called then. It was a herb, but nobody really knew quite how it worked until the adrenergic pathways were worked out. And these adrenergic pathways, of course, came with the discovery of adrenaline. And it was Henry Dale who did a lot of the work on adrenaline to show its effect on smooth muscle in the airways and in the vascular system, and show the polarization of adrenaline on those two structures. But he didn't understand at the time why, until Ulkis described the alpha and the beta receptors, which of course helped explain why there was this dichotomy between the smooth muscle relaxing effect and the vascular smooth muscle constrictor effect. And then as drugs started to be developed, targeting these alpha and beta receptors, we started to get new treatments, and in particular, treatments delivered locally rather than given systemically by inhalation. And this has started at the turn of the 1900s, so this wasn't that new, but obviously the development of the devices became quite a big issue, and these pressurized metered dose aerosols, over which there's been quite a lot of sort of discussion with the climate change and the contents of the CFCs in these. But anyway, these aerosols were developed very effectively, and more selective beta-2 agonists were developed, and as a consequence, we started to get less of the cardiovascular side effects and more of the bronchodilator effects, salbutamol being just one example. And then, of course, what that morphed into these long-acting beta agonists, and now the ultra-long-acting beta agonists, but all playing on the same idea that the alpha and beta receptors were selectively expressed, with the beta receptor being the one in the lung which we wanted to target. And of course, this really brought in the pharmaceutical industry. I only mention one company here, which is Allen and Hambry's, which started off its journey with foods, as you can see at the top, in the early 1900s, but then emerged as a pharmacology company. You'll see those pastils in the tin are cocaine pastils, so already we're getting some sort of quite interesting new drugs coming forward. And David Jack was the person really working at then Allen and Hambry's, as it became known, on salbutamol, and he sort of developed that, and then worked on the long-acting beta agonists with Roy Britton, the salmeterol. Okay. Well, with these beta agonists, everybody thought they had the problem solved. To some extent they did, because it was a very good treatment, as you'll all know. But what started to appear in Australia, New Zealand, and the UK was the increase in asthma mortality that was linked to the overuse of these treatments, but firstly with isoproterenol or isoprenaline back in the 60s, and then a little later on, when everybody thought the beta-2 agonists were safe and people could use them with impunity, we saw another peak going up, particularly in New Zealand, where phentoterol was used as the beta-agonist, a very potent bronchodilator, but also one which produced some significant side effects. And so this was troublesome, a lot of debate at the time, but it came at a time when anti-inflammatory drugs were beginning to be introduced. Now, anti-inflammatory drugs, of course, were a new approach to managing asthma, not just treating the muscle and making it relax, but now getting to grips with some of the underlying inflammatory pathways. And at the time when they were introduced, we didn't know all of that. In fact, it was Philip Hench, working at the Mayo Clinic, who introduced cortisone back in the late 40s for treating arthritis. And he gave systemic cortisone to patients with bad rheumatoid arthritis, and was quoted as saying some of these patients got out of bed and started dancing, having been in bed for 5 years, which was a remarkable observation. But that was in rheumatoid. And of course, we're talking about asthma here, and it really wasn't until the sort of 1952, really, that cortisone and the corticotrophin releasing hormone were being used, ACTH, were being used to actually treat patients with asthma, systemically, yeah. So in a way, it started off on an inflammatory pathway with rheumatoid, but then obviously steroids started to be used quite widely in asthma. And the side effects became very obvious fairly soon. But we didn't have anything else at the time, and certainly when I first came into it, cortisone was still being used quite widely in terms of prednisone and prednisolone as oral steroids. But it was the development, again, by the pharmaceutical industry. I've only given one example here at Allen and Hambre's, GlaxoSmithKline as it became, but also AstraZeneca and other companies. Astra started to make these inhaled steroids. This was actually a skin steroid, beclomethasone. It was designed not to be absorbed from the skin and to be broken down in the circulation fairly effectively, but it was adapted for inhalation. And some luminaries there, Margaret Turner-Warwick at the Brompton Hospital, who was there when Jack Pepys was there, and Romain Pauwels, who sadly died from Ghent in Belgium. They were really pushing these inhaled steroids to control asthma. And so now we were entering into a whole different domain. The last little bit of this historical story is about mast cells and about a non-steroidal anti-inflammatory drug, which is called sodium chromoglycate or cromolyn sodium. And it was Roger Altunian who evolved this because his father brought over this herb called Amnivisnaga, which contained a chemical called chela. And when he heated that at a certain temperature, at a certain pH, it formed what's called a bischromone, which is that structure you see in the middle there, two molecules coming together. And quite by accident, they showed that this could inhibit allergic reactions, in particular the early allergen challenge reactions in the lung, and of course in vitro could stabilize, in inverted commas, or inhibit IgE activation of mast cells. So this was sort of a non-steroidal approach. Roger Altunian supervised my MD thesis when I was at medical school, a postdoc in the 1970s, so I have very fond memories of him. But he was also a little boy mentioned in a book called Swallows and Amazons, which is a very famous children's story in Britain. And you'll see a picture of Roger with the author of that book, Arthur Ransom, with a hat on there, in Windermere, Lake Windermere, where they used to go and have their summer holidays. And this was all about the adventures of a little boy in a boat and going out across Lake Windermere and finding all sorts of exciting things. So that was really where Roger got his sort of inquiring mind from in terms of how he developed chromoglycate. The spin inhaler had its separate origin because Roger worked for the Royal Air Force during the war, repairing and looking at propellers on planes, the Spitfires and the Hurricanes in particular, and then trained pilots during the Second War. And so his interest in propellers drove his idea to develop a spin inhaler, which was quite an interesting bit of the story. Anyway, he set up a company called Fisons. It was in Manchester, just outside Holmes Chapel initially. These are some of his experiments he did on himself. by challenging himself with allergen and putting all these chemicals to seeing if he could block the early reaction. And you can see the number of chemicals that he tested on himself. This was the spirometer he used, which you can see is an old water spirometer there, which is now in the Wellcome Museum in London. Okay. So I suppose that's experimental medicine, isn't it, really? The sort of origins of experimental medicine. But these days, you'd never dream of doing something like that. But in those days, you know, that's how drugs were tried. And the first trial of that particular drug was in 1967. And my mentor at Southampton, the founding professor of medicine, Jack Howell, published that paper in The Lancet, as you can see there, with Roger Altunian, on its effect in treating young people with asthma. But subsequently, as you'll see in that slide, 2003, meta-analyses, when they started pooling all these trials together, showed that its efficacy was marginal, largely because the trials, to be honest, weren't designed as well as they should have been, probably, and they were quite small. And eventually, sodium chromoglycate was removed from the pharmacopoeia, which I think is a bit sad, really. And part of the reason for that is that they were never really able to advance it beyond that molecule, because they didn't know a mechanism. And it's not until relatively recently, I mean 2010, that chromoglycate, the mechanism of its action, has been defined on this G protein coupled receptor 35. And then, as you can see in the last point there, its effect in blocking not only mast cells, but also afferent nerves. And it was known that chromoglycate could block irritant-induced asthma, particularly exercise-induced asthma or cold air-induced asthma. So, you know, a mechanism has appeared, but obviously too late for developing that. Finally, we come to this enigmatic molecule, slow-reacting substance of anaphylaxis, that was described by Henry Dale, and they had no idea what it was. But in fact, Charles Kellaway and Trethewey, working at the Eliza Hall Institute in Melbourne, were the ones that really put this on the map. You can see the slow reaction there compared to histamine, where it was difficult to wash out the SRSA, hence it was given this name, slow-reacting substance of anaphylaxis. Well, what happened then, of course, was the definition of what these molecules were. A huge race. When I was in Boston in 1978, I can't describe to you the competitiveness at that moment, of groups around the world trying to get the structure of SRSA. And of course, it was Bent Samuelson, really, who sort of came up with that eventually. But K. Frank Austin, working with E.J. Corey in Boston, and I'll just sort of ring them because they were quite amazing people. Those two got the Nobel Prize, not just for this, but Corey for his structural elucidation of cyclosporin, I think, and Bent Samuelson because he'd worked on eicosanoids beyond the leukotrienes prior to that. With John Vane, they'd worked on prostaglandins. So this was a sort of eicosanoid Nobel Prize award period, which was quite a nice period. But of course, it turned out that these were cystenyl leukotrienes that were causing this slow-reacting problem in the airways, and of course drugs were developed, as you can see there, particularly the montelukast, which is still used today, the oral drugs. And Jeffrey Drazen, particularly, and my good friend who's also passed away recently, Tak Lee, worked together to try and sort of map out some of the pharmacology of these cystenyl leukotrienes, which they did very effectively. So that brings me to K. Frank Austin, who I worked with in 1978 to 1980, in Boston. Then it was the Robert Brigham Hospital, but it's morphed now into the hospital where Bruce currently works, the Women and Brighams. And K. Frank was part of that evolution, really, as that hospital started to get larger. He died 2 weeks ago at the age of 95. A remarkable man, father of immunopharmacology, really, to be quite honest, in our field. Again, I won't read some of that. It's obvious to you what his contributions were. But he trained over 200 postdocs that became professors across the world, in almost every country of the world. So this wasn't just about research. This was about his ability to really train a world-leading group of scientists that have endured over half a century. And that is absolutely an amazing achievement. What's also interesting is that he did his postdoc with Henry Dale at Mill Hill, and he started working on SRSA in those early days, trying to sort of unravel what it was all about. And so really, when we sort of look back on all of this, we can think about the inflammation starting to really take up a positive position in the asthma paradigm now. And I won't read all that. You can see the historical bits here. But it was about trying to understand the individual cellular components. It was about understanding what chemicals they released, whether they be mediators, whether they be chemokines or cytokines, and putting it together in a way that might reveal new therapeutic opportunities. So Al Sheffer, who also was working in Boston, and Bruce will remember Al very clearly, I'm sure, but he was more the clinical end of K. Frank Austin's enterprise there. He chaired with Claude L'Enfant and Suzanne Hurd from the National Institute of Health this amazing first document, which really put inflammation at the heart of asthma. So now, instead of just talking about bronchospasm and smooth muscle, we were talking about inflammation and control versus relieving with these different treatments. Control with inhaled steroids and chromoglycate, relieving with bronchodilators. So, a story started to emerge that was very attractive. Also, we got a bit braver. I had a postdoc from New Zealand who one Saturday morning slipped a bronchoscope down an asthmatic and took some biopsies, and Richard Beasley, as his name was, he's really quite famous now, but these are some of the pictures that were taken from those biopsies in those days. And my good friend Ratko Djukanovic and Peter Bradding and Susan Wilson were all part of this. But you can see the eosinophil in the center there and the mast cell on the left, and also this remodeling, this matrix and the myofibroblasts in the center, and the epithelial damage, and this rather unusual form of eosinophil degranulation that became quite an interesting research area in its own right, shown on the right-hand side. So this was actually putting the inflammation in context, and this context was, of course, an altered structure. The muscle had changed, the epithelium had changed, the underlying mesenchymal nerves had changed. And it was this idea that the structural changes were being informed by some sort of impaired wound healing response, that the epithelium was playing quite an important role in this, in orchestrating not only a sort of healing by secondary intention with fibrosis, increasing muscle, increasing nerves and blood vessels, but also sustaining the inflammation now, because the epithelium wasn't repairing after the injury, and in severe asthma this was a problem, and part of that turned out to be the fact that the epithelium itself was primarily abnormal in this disease. So that was the next level, really, beyond the inflammation, that we're in now to the epithelium acting in terms of programming the underlying responses. And of course the epithelium was the substrate of all of these things in the environment that stirred up asthma, like viruses and air pollution and so on. So in a way now we had a way of understanding how that environment out there was actually feeding into the asthma process beyond allergen exposure. And I won't go into details of all of that, but in essence the epithelium seemed to be quite critical in all of that. And it was critical because this structure was abnormal in asthma, but not only was it abnormal in asthma, but when they started doing all this genome studies, they started to discover that a large number of the susceptibility genes of asthma resided in this epithelium. And of course that's an important part of the story, because the epithelium now is serving as a barrier, and it's this barrier function that was impaired. And now we know there are hundreds of genes that are abnormal in this epithelium in asthma, so it's not just a handful of these. And so the story really that's built up over this period of time came to the point where we have multiple environmental factors feeding into a susceptible airway set up by the genetics, making the immune system hyper-aware, such that when allergens started to come in early life, we got sensitization and the asthmatic process taking hold, and that's kind of summarized in that slide. And in a way, it's that knowledge that's enabled us to move on to this next phase that my colleagues, I think, will talk about in a minute, which is really thinking of asthma not as a homogeneous entity, but as a disease with different molecular players that change in accordance with individuals' clinical expression. And you'll be aware of the different subtypes of asthma defined by the immune response, and I've just summarized them briefly on this slide. But what that has led to is the design for the first time of molecules targeting the underlying chemical mediators that are actually driving these inflammatory reactions. And you can see the various monoclonals that are directed to various subtypes of asthma there. I mean, we're not there yet. This is just the beginning, but it's a very exciting development as we go forward. And I just want to end, really, on one point, is that we've concentrated, I think, in my lifetime, really, on how we understand asthma as a disease and how we might intervene in it once established. But the big issue here is why it's there at all. Because as I said at the beginning, Salter, it took him 15 years to find these 20 or 30 patients, and yet 1 in 4 children in our schools carries an inhaler now. So something has happened to the environment over this time. And it's beautifully pictured here by these studies from Finland. There are many, many other ones to show what's happened since the 50s and 60s to the prevalence of asthma and of allergic diseases generally. And this rise in asthma and allergic diseases is all around the environment around us. So I think as we look to the future, we've really got to start thinking about this and start thinking about how can we stop asthma and allergy happening in the first place, instead of waiting for it to happen and then pump loads of drugs into these people. And I think there are some clues that are coming out now, and I've listed them there, because we know in the environment that there are very, very many different factors, but they're all pointing in one general direction. The first things that damage the lung, and you've got chemicals and pollutants and viruses and all of that there, but you've also got this lifestyle thing, which is all about what's happened to our world around us in terms of what we're exposed to early in life. It used to be called the hygiene hypothesis. Some people got quite cross by that term, but David Strachan initiated that term. We've gone quite a long way beyond that now. And I think what we're really talking about here is the loss of microbial diversity in our environment, both in the gut and in the lung and skin. And that this diversity loss is what's leading to the absence of the early life programming or the innate learning of the immune response, which is leading to this rise in allergy that we've witnessed in the last 30-odd years or so. So I think as I sort of leave my medical career and immunopharmacology career, what I'd like to see is some understanding of this, okay? Because this has to be accessible. This is acquired. This is the environment. And we know that, kid is brought up on livestock farms and things, and with beautiful work in Germany and the United States with the Amish and Hutterites. We know that these farm environments can be protected, almost totally protected. So, yeah, we can't kind of expose everybody to cows and give them raw milk to drink, but there has to be something in that story that we can harness in some way. And it's about really getting our resilience set early in life, such that when we're confronted by all these different things like viruses and allergens, our resilience can actually keep it suppressed, and allergy and asthma stops occurring. Thank you very much.
Thank you, Professor Holgate, for just a marvelous initial lecture at this session and highlighting the important contributors conceptually, and also the individuals who have contributed so importantly as our heroes for those of us who are working in asthma, yourself included, in this journey. And now we enter this syndromic phase of asthma where we're endotyping and stratifying and trying to identify specific targeted treatments. We have time for maybe 1 or 2 questions. If somebody would like to come to the microphone. Please, microphone 2 and then microphone 4.
Hi, I'm Amanda from the University of Calgary.
Hello.
I was wondering if you had an opinion on why antihistamines were not as effective in those cases of asthma as the mast cell stabilizers. What were they doing differently in those cases?
Well, yes, that's a very good question. And Henry Dale kept asking that question because he thought he'd got his antihistamines wrongly designed. But we all know now that antihistamines, they may have a small effect on certain parts of the allergic response, the early response. They can have a small effect on the early response. But I think mainly because the chemicals coming out of all of these cells, in terms of the target tissue, probably aren't responding that, vigorously to these mediators. I mean, in a way, that's why SRSA sort of made its appearance, because it was that bit of the contraction that nobody could really explain. And it would seem that SRSA is probably more relevant there. And there's going to be other contractile agents there as well. This was way before the sort of neuropeptide era too. So this amplification process where neuropeptides are released in a sort of chronic wound scenario adds to that amplification response. Yeah, so I think we've dropped antihistamines away. But of course in rhinitis and in skin disease, they're pretty good. So it's not just, it's just asthma, I think, is the problem.
Thank you.
Yeah, Mike Parnham, Epiendo Pharmaceuticals. That was really nice talk. Thanks very much. It's nice to important to point out where we've all come from. I was interested in your list of factors affecting the environment for development asthma and allergy, and including oily fish intake. Is that related to the modification of the microbiome, or is it also related to the generation of specific resolving mediators?
Yeah, well, absolutely. And I would say in answer to that question, both. Because there's no doubt that some of the polyunsaturated fats in particular that are obviously present in oily fish can have beneficial effects on the microbial population in the guts. And of course it has gone into trials acutely, but there've never been any what I call lifetime trials yet over the life course. So we don't really know that if you gave these things right from the beginning and look at people at the age of 25 or 30, whether in fact, it would have an effect. But I think the resolving mediators are really fascinating in their own right, and I haven't touched on them at all today, and I apologize for that. But yes, I do think they're very important as well. And when we had the previous question about various additional mediators, I mean, leukotrienes and prostaglandins and histamine are only one set of mediators, but we've got other mediators which are relaxing in nature and other ones that are contracting in nature. And I suppose it's a competition between all of this soup of chemicals in the lung that gives rise to some of the different clinical phenotypes that we see of the bronchoconstrictor response.
So it's good to keep eating Scottish salmon.
Oh, definitely, yeah, definitely. And the other thing, I do feel that clinical trials now, we think of them of 6, 8, 10 weeks, 2 years, 3 years. But, what we're talking about here are lifetime effects. And this is the life course. And maybe we should start thinking rather differently about how we set things up and what we do over time. And certainly, when you look at things like the Biobank and other collections where they're doing these long-term studies, it's really very interesting to see new pathways emerging. Thank you.
Yes, maybe as we've moved away from cod liver oil treatments in children. One final question, Steve, if you might.
Rob Newton, University of Calgary. So wonderful talk, Steve, and fantastic historical perspective. Observation, very white, very male.
Yeah, totally.
Moving forward, maybe we get to have a more diverse outlook, which would be great. As a child growing up in the 1960s and 70s, I remember the spin inhalers as being horrible things to take and not very effective. When Ventolin switched from tablets to inhalers, that was a fantastic step forward. But personally, asthma was only really controlled for me once I started taking inhaled glucocorticoids or corticosteroids, which would have been, I don't know, 1970s or so.
Yeah, that's right.
So the question, and I work on glucocorticoids, as a number of people know, what do you see the future of glucocorticoids in the context of asthma therapy? They control mild to moderate disease fantastically well. So is it worthwhile exploring those fringes to further improve glucocorticoid therapy? Or is it a done deal and we now just focus totally on the biologics and those other things?
Well, I mean, to be honest, you're the best person to answer that question because it's your field of interest. I mean, I think corticosteroids, were thought when they came in as inhaled steroids to be a panacea for asthma. And of course, they've gone right across the therapeutic severity range. And if you look at the guidelines for asthma over the years, it's reflected that. But I think what we've ended up with now is an interesting situation where very high doses, either given inhaled or systemically, but certainly by inhalation, may be altering the airway wall itself. In other words, we may, by giving very high doses of these things over prolonged periods, be altering the very organ that we're trying to actually suppress in terms of the disease process. And I've always had a kind of sneaking feeling that, enough is enough. Too much is too much. And I think pharmacology has tended to follow that sort of paradigm in other disease areas as well, that there becomes a limit to what you can achieve with certain things, no matter what you do with the shaping of the molecule and changing it. And that, I think we need to be thinking much more now about targeting those pathways that don't seem to be suppressed by steroids. And of course, these alarmin molecules that are being described coming out of epithelial cells and mast cells, which I think we'll hear about in a minute, they are becoming an increasingly interesting area to start thinking about. But also, Charcot-Leyden crystal, that's a galectin 10. I mean, that molecule itself can be targeted. And certainly research work that's come out of Belgium recently would suggest that monoclonal antibodies that stop the aggregation of the galectin 10, which is the lysolecithinase that comes out of the eosinophil, has therapeutic effects. So there are some things that might actually still be there that we're looking at, but we haven't yet got the therapeutics properly aligned to target them. Thank you very much. Good. Well, look, thank you very much for those questions. We're going to go straight on now, and I'm going to introduce my friend and colleague, Emily Swindle. Now, Emily works in Southampton, and I was very pleased to be able to recruit her, I think. I don't know whether that's the right word, Emily. Anyway, she decided to come to Southampton after leaving the NIH, where she was working on mast cells there, and has spent a very productive time with us. And she's going to tell us a little bit more about mast cells, and I think touch on remodeling as well. So thank you very much, Emily.
Excellent. So I want to thank the organisers for giving me the opportunity to talk about my research today, and to be able to follow such an eminent professor as Stephen is a real honour. So I hope I can keep up the standard of the talks that have gone before me. Okay, so I'm going to talk about the epithelial-mesenchymal trophic unit within asthma as a druggable pathway. And as Stephen's already alluded to, obviously that concept started in Southampton back in the 2000s, and it's really been developed and pushed forward over the time. So, again, asthma is a real challenge. We've already touched on that already with what Stephen has talked about. And it is a complex disease involving chronic inflammation as well as structural remodeling. And patients with severe asthma have poor symptom control, have more exacerbations and comorbidities than patients with mild or moderate disease, despite that increased therapy of inhaled corticosteroids or maybe tablets. It is now a heterogeneous disease, that's no doubt that it is. Asthma is now more of an umbrella term for a syndrome. And it has distinct phenotypes, so whether that's mild, moderate, severe, if it's type 1 or type 2. But by understanding the endotypes of asthma, those are the mechanisms driving disease, we can maybe look at stratifying patients and developing new therapies. And there is a real unmet need for new therapies, especially in severe asthma, where patients experience severe exacerbations, life-threatening, and death resulting from that. Obviously, in Southampton, I have to talk about the epithelium. And we know that it's a key driver of development in asthma. As Stephen already alluded to, there has been various genome-wide association studies that have identified a large number of asthma susceptibility genes localized to the epithelium. And through bronchoscopy and other techniques of collecting samples from patients with asthma and running through different molecular techniques, cluster analysis has, of the bronchial brushings of patients with asthma, has identified distinct phenotypes. And work done in Southampton before I arrived has shown that there is a functionally defective epithelial barrier in asthma, as well as a deficient innate immune response with reduced interferons and increased TSLP and other pro-allergic factors. But this is what I really want to focus on today. So this is kind of an updated slide from what Stephen showed. And really the key point of this is just to demonstrate fundamentally that the airway barrier in a normal situation is exposed to a whole range of noxious substances. So in healthy individuals, the airway epithelial barrier can resist those perturbations. And so the epithelial barrier, through the expression and polarized release of factors, actually controls the tissue microenvironment. And there are three epithelial barriers that we talk about. There is the physical barrier, which is the polarized expression of tight junctions, which control the paracellular flux of ions and macromolecules. There is the chemical barrier, with the excretion of mucus and other factors that can trap pathogens and noxious substances, inactivate them. And along with the cilia, the mucociliary escalator is activated and we can remove those noxious substances from the lung. But if the physical and chemical barrier are compromised, then we get activation of the immunological barrier. And the epithelium has a variety of receptors that can be activated by various pathogens to mount an immune response, which in a healthy individual would lead to recruitment of the cells and expelling and reversal of that inflammation. However, in patients with asthma, there is dysregulation. Why is it that patients with asthma are more susceptible to these environmental challenges? So whether that's a virus or pollen or smoke or pollution, the airway of the epithelium, the epithelium of the asthmatic is far more sensitive. And we can explain that by three mechanisms: that there's injury to the airway epithelium in asthmatics, and they're more susceptible to that injury from noxious substances. And it results in dysregulation of that epithelium and compromise of the barrier. And we know there is disruption in those pathways. There's the release of pro-inflammatory mediators, the dysregulated release that instructs many cells, whether these are resident cells such as dendritic cells, or the resident mast cells and ILC2s that drive type 2 inflammation. And what I really want to point out is that the resident ILC2 and mast cells can be activated straight away to produce type 2 cytokines, where the dendritic cell has to go away and tell a T cell what to do, and that has to come back and release those cytokines. So there's dysregulation in inflammatory mediators, which has a direct effect on the epithelium that means it can't repair itself properly. So there's an aberrant repair, so that when we get re-exposed, that actually that barrier is already compromised. There is a remodeling response, so the airway epithelium communicates with the underlying mesenchyme to activate fibroblasts and airway smooth muscle to remodel that airway. And we, in asthma, see a lot of those factors that obviously Stephen's talked about. There's an increase in the reticular basement membrane thickness, deposition of collagen and extracellular matrix. There is increase in goblet cells, There is airway smooth muscle hypertrophy and hyperplasia, as well as remodeling and formation of new blood vessels that are more permeable and edema. And so what is going on is obviously that, looking on the left there from a normal healthy airway compared to the right of an asthma patient, you can see the extent of that remodeling that's going on. And it's on the background of that patients are responding to the airway microenvironment with different environmental factors. And when I'm talking about the airway epithelium and its role in responding to environmental factors, actually it's the genetic predisposition of individuals which exacerbates that. So it's not just about the environment, but it's that gene-environment interaction. And so there are genes associated with how the epithelium responds to environment. So for example, we've got CDHR3 there, which is the rhinovirus receptor for rhinovirus C. We've got genes that are involved in repair and resolution, such as TGF-beta. ADAM33 is another one that I'll touch on. And then also genes involved in dysregulation of innate immune responses, such as IL-33 and TSLP. And it's not the fact that you have just a mutation in one of these genes, but there must be a combined effort of a mutation in a gene along with an environmental impact that together contributes to the disease progression, whether that's in the early life or develops later. And there's been various associations and trying to get functional effects of these mutations to see whether it actually results in a functional effect. So what I'm going to talk about briefly is to look at some different responses in the EMTU to highlight some druggable targets. So I'm going to touch on the interferon beta story, but also defects in the innate immunity, such as IL-1 alpha and IL-13. Defects in barrier function, I'm going to focus on interleukin-33 here, which is epithelial-derived cytokine. Talk about EGFR and a role in steroid resistance in neutrophilic asthma, and then touch on the ADAM33 gene and its effects with bronchial hyperresponsiveness. So, an example of epithelial damage is induced by human rhinovirus. So, we are aware that over 80% of asthma exacerbations are associated with respiratory virus infections, with over 2/3 of cases associated with human rhinovirus. We know that patients with severe disease have more severe lower respiratory tract symptoms following infection, which is strongly correlated with viral load. And there is a deficiency in the innate immune response to asthmatic epithelial cells, where they produce less interferons, and by doing so, there is more virus in those airways. And specifically in severe disease, we don't normally see this in patients with mild. And while there are over 100 serotypes of rhinovirus, there are no effective vaccines or treatment. But the observations back in 2005 from Southampton that showed that there was a deficiency in interferon beta in asthmatic patients resulted in a spin-out company called Synergen that developed inhaled interferon beta therapy that was subsequently sold to AstraZeneca, and a trial which was published back in 2014 by the team showed an effect. So just on the left-hand side is the severity of asthma on the x-axis, which step 2 is mild, step 3, and step 4 and 5, where 4 and 5 is severe disease. And we see there's a change in asthma control questionnaire when patients have acquired a natural rhinovirus infection. And what this study did is within 24 hours of patients having symptomatic symptoms of a viral infection, they were brought in and given intervaled inhaled interferon. And on the right, what the graph is showing is that there was a reduction in their asthma control questionnaire. So 0.5 is a clinically significant difference. And so they were symptomatic, and only in those patients with severe disease where interferon was administered, that was reversal, showing a true effect. So in respiratory viral infections, targeting interferon beta is a good therapy. But I just wanted to switch to mast cells for a moment, because we know in asthma, especially in severe disease, that mast cells associate with the bronchial epithelium. And we showed back in 2017 that mast cells are susceptible to rhinovirus infection. So unlike other innate immune cells that would respond to rhinovirus, mast cells were unique in terms of they actually produced active virions, infective virions. And so while we could demonstrate that interferon beta can protect mast cells from rhinovirus infection, this may not be enough in severe disease where there is a deficiency. So mast cells in that situation may be the reservoir. And we know that in patients with viral infections that there is an increase in the epithelial-derived cytokine IL-33, and it's associated with severity of exacerbations. And so we wanted to look at the crosstalk between the two, because we know that IL-33 or its receptor, ST2, are asthma susceptibility genes. And they are associated in the bronchial epithelium and again increase the disease severity. In terms of mast cells, they respond to IL-33, but they also can process it to a more active form. And a beautiful study a few years ago now demonstrated that in severe disease, mast cells and basophils are the target for a splice variant of IL-33 that is increased in severe asthma. Mast cells can respond to IL-33 as well. So thinking about severe disease where we have increased IL-33, deficient interferon, and mast cells that can contribute to that viral load, what is the influence of IL-33 on mast cell function during viral infection? And what we found, we did a transcriptomic analysis, so we exposed mast cells to IL-33 and ran an RNA-seq, and what we showed that the IL-33 induced an antiviral signature. So there are over 400 genes that were differentially expressed, and when we did pathway analysis, we could see that on everything highlighted in red are pathways associated with viral immunity. And specifically, the influenza pathway pulled up, and there were lots of genes in that pathway, so suggesting that IL-33 is a good thing. And we verified some of these genes. So, for example, MDA5, the gene IFIH1, which the rhinovirus is sensed by, was upregulated by IL-33 treatment in mast cells. So we decided to expose mast cells to IL-33 and then challenge them with virus and see kind of what could we enhance this antiviral activity. But actually what we found was the reverse. Paradoxically, we've seen that virion release was increased when mast cells were exposed to IL-33. And we demonstrated this was mediated by ICAM1. So this is the receptor that rhinovirus 16 uses for entry. And we could see that IL-33 increased that receptor. And if we blocked that receptor with an anti-ICAM1 antibody, we could reverse that increase in virion release. So this suggests that in the context of severe asthma, that IL-33 actually contributes to that exacerbation directly by affecting mast cells, although obviously this has to be demonstrated in vivo. We also showed in the RNA-seq data, and we haven't published this yet, but there are remodeling factors that also were increased. So the collagens, collagen 5 and 6 were increased, as well as matrix meloproteinases, showing that potentially mast cells can contribute to that remodeling. And there's a lot of evidence out there that mast cells already do that, but not in the context of virus infection. So overall, we kind of summarize this on a slide here, so that following rhinovirus infection in asthmatics, which already have a deficient interferon response, there's release of IL-33 that can increase the ICAM1 expression on mast cells. that can produce more active infectious virions, and in this way mast cells may contribute to the exacerbation. So that's kind of looking at IL-33, but it's not just in severe asthma which linked to viral infections where IL-33 is upregulated. But in another study, Jean-Marie Perraudet, who came over from France to work in Southampton, was interested in gastroesophageal reflux disease, because in severe asthma, there are lots of comorbidities, and one of those is reflux. So she developed a model where she exposed air-liquid interface cultures of epithelial cells to a mixture of pepsin, acidic pH, and bile acids, and looked at the effect on the epithelium. And what she demonstrated, there was disruption in the barrier integrity, so this macromolecular permeability was increased, and in severe asthmatics, it was significantly different from healthy patients, showing they were more leaky, shall we say. She confirmed that actually in those cultures, IL-33 was upregulated. And the key thing was to take bronchial biopsies from patients with severe asthma and GORD, and actually could demonstrate that the IL-33 was localized to the epithelium. So this is a different type of asthma, also demonstrating IL-33. So then that comes to, do these therapies be effective in severe asthma? And the short answer to that, while studies are obviously ongoing, there are anti-IL-33 therapies where we're still awaiting the outcomes of trials. But for the anti-ST2 trial, it's published back in 2021, that it was effective at reducing annualized annual exacerbation rates in patients with asthma. And if you look on the right-hand side, if you stratify patients based on eosinophilic count, so patients that have higher than 300 cells per microliter, it was not as effective as in patients with low eosinophilic count. So it could be a targeted therapy for patients with asthma, which are not eosinophilic driven, but neutrophilic, which is that difficult to treat severe asthma. And just to kind of throw something in about mast cells, so we know that if we look at patients with severe disease, there is a mast cell signature which is IL-33 related. And if we look at if we enrich for the different types of inflammatory cells, that it is IL-33 activated mast cells that are enriched within the neutrophilic or mixed eosinophilic, neutrophilic severe disease. So kind of thinking that it's not just one thing of an IL-33, but there are immune cells we need to think about, and maybe that actually putting the two together could help in this severe neutrophilic disease. So that's IL-33. I'm now going to talk about IL-13. So IL-13 is typically from type 2 inflammatory cells, but there is a role in a demonstration that IL-13 can also come from structural cells such as the epithelium. And this is a mouse model from Donna's group in collaboration with Hans-Michael Hatchie. And Lizzie kind of used a IL-13 expressing mouse, so you can induce IL-13 expression, and what we demonstrate is you can get bronchial hyperresponsiveness in that model, and the bottom graph is just showing that it's a mix of neutrophilic and eosinophilic inflammation. And it's steroid-resistant neutrophilic inflammation. So on the top bar is just the mouse IL-8, shall we say. And we can see with the dexamethasone, it is not inhibited, but with an EGFR inhibitor, it is inhibited, that AG148, and it conversely reduces the amount of neutrophils in that disease. That's great in the mouse, but mice, as we know, aren't humans. But the key thing they did was use the eubriopred cohort to look at the epithelial brushings and look at the signatures within that cohort, which is a mixture of healthy, mild, moderate, and patients with severe disease, to do non-hierarchical clustering and TDA analysis. And what they showed was the bronchial brushes from severe asthmatic patients. There was a subset that had neutrophils and EGFR ligands demonstrating that this could be translated. So again, IL-13 treatment hasn't really panned out in terms of anti-IL-13, but maybe in severe neutrophilic asthma, there may be a role for it there. So I just want to switch to IL-1 alpha. So this is a role in exacerbation. We have in vitro models of the EMTU where we have airway epithelial cells and fibroblasts in a polarized model. And what we've done is expose them to double-stranded RNA. And you can see in the core culture, there's enhanced mediator release. We also see disrupted barrier, and we can block it with IL-1 receptor antagonism, and this was published some time ago. This is with double-stranded RNAs, a mimic for viral infection, but we also see it with polyl-arginine, which is kind of a mimic for eosinophilic cationic protein. And in exacerbations of asthma, we see increase in IL-1 signaling, and while the studies of anakinra are kind of mixed, we'd say that I would think about reconsidering that in terms of exacerbations of disease. My last slide I just want to talk about is ADAM33. So we know that ADAM33 is an asthma susceptibility gene. It's expressed in the mesenchyme. It's a transmembrane protein that's cleaved to a soluble form that's released into the airway. And it has multiple effects on remodeling. In a mouse model, again, Lizzie Davies and Hans-Michael Hitchie, they could induce soluble ADAM33 within the mouse model. And what was a key observation is that we don't get inflammation, but we get remodeling in that mouse model. And actually, it is reversible. So targeting soluble ADAM33 as a therapy may be beneficial. The other thing that they demonstrated was actually soluble ADAM33 augments airway inflammation, but it required lower doses of allergen, so lowering the threshold for sensitization. So this is an example of, obviously, a mesenchymal-specific gene influencing the tissue microenvironment. So this is my last slide, just to kind of pull it all together to say that we need to consider, obviously, the genetic predisposition of patients with asthma. But it's absolutely pivotal to think about the environmental exposures that impact the airway epithelium and how it talks with the underlying mesenchyme. And it's really that combination of the environment and the genes that will ultimately be the targets of new therapies coming through. And I've touched on a few of them, but you can see there are very many more other targets that we can consider. So I'd just like to have acknowledge people that have contributed, my funders, and I'll just be more than happy to take any questions. So thank you for your attention.
Thank you very much indeed, Emily. So we've probably got time for one question, just to make sure. If there's time at the end, then we can come back and maybe ask things, but we'll just keep things on time. So is there any question anybody wanted to ask? Yes, please, on microphone
I can ask
two.
a question. So Emily, just a technical question actually. If I am not wrong, IL-33 is one of those cytokines that is processed into a small, and I think you said that.
Yeah.
So my question is very simple. When you show the immunohistochemistry and the data, are you looking at the processed version or the full-length version, which would be less biologically active?
Yes, so that's really interesting. So obviously IL-33 does get processed from a reduced to an oxidized form, and it's not really clear how the antibodies are detecting that, because a lot of them will detect the reduced form. And it's really, you know, that oxidized form that I haven't touched on, but actually kind of drives remodeling responses as well. But we know it's been demonstrated that the anti-IL-33 therapies that are out there, they do prevent the oxidation of IL-33. So they could hit inflammation as well as remodeling. But yeah, it's not very clear.
Good, thank you. Thank you very much indeed, Emily. We'll move on now to our next speaker, and I'll hand over to Bruce.
Thank you. Thanks for that wonderful talk. Our next speaker is on oral communication for an early stage investigator. It's really my pleasure to invite Amandah Necker-Brown from the University of Calgary to the podium, and she'll be telling us about her work in inducible gene expression of IKKε that's dependent on NF-κB signaling in human pulmonary epithelial cells. Welcome, Amanda.
Thank you so much. See if that works. Okay, perfect. So yes, my name is Amanda, and I'm very humbled to be presenting amongst such amazing speakers. And today I will be presenting my master's project. So I'm a pandemic-born graduate student. This is my first opportunity to present at a conference. So I'm glad they put me in the most giant theater ever. And I'm going to be presenting my project involving the regulation of IKKε as it relates to the regulation of inflammation and inflammatory signaling cascades. So, as discussed before, asthma is generally characterized by chronic inflammation of the airways, and especially discussed by Dr. Swindle, inflammation is not well controlled in patients with severe asthma by glucocorticoids. So now, although severe asthma only accounts for up to 5% of individuals with asthma, it does account for over 50% of the economic costs associated with asthma. Thus, understanding the function and regulation of pro-inflammatory genes that avoid full repression by glucocorticoids, which I will explain is relevant for IkB-KINASE epsilon, may provide targets for pharmacological interventions or biomarkers in severe asthma. So why do we want to study IkB-KINASE epsilon? IKKε epsilon is highly induced by inflammatory signaling, as I will show you later on, yet it's not fully repressed by glucocorticoids. And it is implicated as a pro-inflammatory gene in a lot of different cell types and diseases, but its role in regulation in airway epithelial cells is largely unclear. So it has been implicated in other inflammatory diseases like arthritis. It's as well a potential inflammatory target in neurons. It's also implicated in pulmonary inflammation, but a lot of it has been studied in neutrophils, but not in airway epithelial cells. So, as a bit of an overview, inflammation is mainly driven by cytokines like IL-1 beta and TNF that bind to cell surface receptors to activate intracellular signaling cascades through the induction of gene expression. Now, all of these IkB-kinases are essential to that signaling cascade. And we have the canonical IkB-kinases, IKK alpha and IKK beta, which are known to activate NF-κB. But on the other side here, we have the non-canonical IkB-kinases, including my favorite IKK epsilon and TBK1. Now, these are more known to activate other transcription factors known as interferon regulatory factors. So, as discussed, airway epithelial cells are very important for asthma. They're the first target for inhaled insults, and they're a key driver of inflammation in asthma. So that's where I'm going to be looking into the expression in. So they are also, I will be using the models of A549 cells, Bs2B cells, and primary human bronchial epithelial cells, grown in both submersion culture, just like our cell lines, but also at airway liquid interface. So as Dr. Swindle was showing as well, that's when just the basolateral surface of the cells are exposed to media, so it allows them to differentiate into a lot of different cell types, more closely mirroring a physiological epithelium. And I will be treating these cells with IL-1 beta and TNF to model inflammatory gene expression and signaling in airway epithelial cells, and then using these steroids, glucocorticoids, dexamethasone and budesonide to identify the effects of glucocorticoids on gene expression. So jumping into the data here, I was looking at the expression of all those IkB-kinases in airway epithelial cells using our lab's RNA sequencing data in A549 cells. So as you can see, initially, all of the other kinases are quite highly expressed in these cell types, yet IKK epsilon is quite lowly expressed. However, when we add inflammatory stimuli, it becomes quite similarly expressed to all of these other kinases, suggesting that it could play a role at these time points. So we've also looked into them in those airway liquid interface cells, and you see the same upregulation. So compared to the non-stimulated condition in gray, adding in red IL-1 beta, we get this extreme upregulation, where it becomes similarly expressed to these other IKB kinases. Importantly, at this 6-hour time point here, you can see that IKK epsilon is, even though it's highly upregulated by inflammatory stimuli, it's not repressed by glucocorticoids. So I looked a little bit more into the mRNA expression using qPCR, and you can see that yes, at 6 hours following inflammatory stimuli, we don't get a significant reduction when we add glucocorticoids. So this really shows that IKK epsilon could be playing a role in even in patients that are taking glucocorticoids, and because it's known as an inflammatory gene, this might be important. And at 24 hours, just like you see in the sequencing, although there is a significant effect of the glucocorticoid, it's nowhere near drop down to basal levels. And as well, the protein expression mirrors these same trends in both A549 and ALI conditions. So my hypothesis, because IKK epsilon was induced by inflammatory signaling, was that the canonical NF-kappa B cascade over here promotes the expression of IKK epsilon. And this may allow for potentiation of that signaling event, or allow for other signaling molecules to come in and activate IKK epsilon at other time points. And that's what I would be doing for this project, is interrogating the role of NF-kappa B in the induced expression of IKK epsilon. So to start with, I used 3 canonical IKB kinase inhibitors, so inhibiting IKK alpha and beta over here. And they were all able to significantly reduce IKK epsilon gene expression. Secondly, I used IKB alpha delta N overexpression adenovirus vector, which basically overexpresses a truncated form of IKB alpha that will hold P65, that like canonical NF-kappa B subunit, in the nucleus, so it can't bind DNA to initiate gene transcription. And that was also able to significantly reduce IKK epsilon mRNA expression. Thirdly, I used P65 siRNA to knock down P65 protein, and then again, we get that reduction in IKB kinase epsilon expression. Looking into both our labs, chip sequencing data in A549 and Bs2B cells, which, if you're not familiar with chip sequencing, it basically shows where proteins are binding within the genome. You can see that the IKK epsilon locus, shown above here, there's 3 different regions where both P65 is binding not in non-stimulated conditions, but increased with that IL-1 beta stimulation. And similar in Bs2B cells, we get those same 3 regions enriched in those conditions. And interestingly, in those same spots, we also see polymerase II binding, showing that these could be important for gene transcription. So because we saw that P65 was binding these sites, I wanted to clone these into a luciferase vector to see if they could actually drive transcription. So these are the same chip sequencing peaks that we saw before. The R1, R2, and R3 regions, which I'm going to name them. And I cloned them all separately into this luciferase plasmid, so instead of P65 binding to IKK epsilon, it's binding to the luciferase promoter and driving luciferase gene expression. So first, I'm just showing that R1, R2, and R3 were all able to significantly induce gene expression when exposed to IL-1 beta and TNF. And then I just wanted to show that also that regulation was dependent on that canonical NF-kappa-B cascade. So I added back in those IKK inhibitors, PS1145, ML120 beta, and TPCA, and they were all able to significantly reduce that luciferase activity. And as well, I added back in that IKB alpha delta N overexpression virus, significant reduction. So to really look into what sites are actually driving this expression, as you can see, I have labeled the JASPAR NF-kappa-B binding sites. So you see 3 in this region over here, 1 in region 2, and 2 in region 3. And I went back to the clones that I had already made, and then I used site-directed mutagenesis to take out all of these regions. And as you can see, when we remove both S1, S2, and S3, even separately, they all have a significant effect on luciferase activity when exposed to IL-1 beta and TNF. And when you take out all 3 of them together, as you can see what I did here, it completely ablates that induction. So this just shows that all 3 NF-kappa-B sites are important for full transcriptional activity. In that second region there, although this was a pretty weak NF-kappa-B binding site, it barely looked like one when you look into the actual sequence. I tried to delete it and nothing happened. So this does suggest the existence of non-canonical NF-kappa-B binding sites. Potentially P65 could be binding with another factor so that those sites are looking a little bit different. And then in that third region over there, the R3 region, compared to the wild type, when I deleted the S5 region, we didn't see any effect on induction, except that S6 region there did have a significant effect. So this was nice because it confirmed that the necessity of NF-kappa-B binding sites correlated well with the JASPAR binding scores, because the S5 had a very weak NF-kappa-B binding score, so it didn't look like a canonical NF-kappa-B site, whereas S6 was a stronger site, so it checks out. So in conclusion, IKK expression is dependent on early NF-kappa-B signaling. And important, this induced expression is not fully prevented by glucocorticoids. So whatever IKK is doing, it could be doing this in cases of severe asthma. So what are the implications? IKK has the potential to potentiate a signaling in response to these same stimuli, IL-1 beta and TNF, that are going to be quite widely expressed in chronic inflammation. Or, because we don't yet know if IKK is activated once it is induced, it could prime the cell for alternative stimuli, such as viruses or bacteria, to come in and activate IKK and kind of prime the cell for those alternative responses. Interestingly, how Dr. Swindle was talking about those viral exacerbations, IKK is implicated in viral signaling, so this could be a potential mechanism for that. It also could contribute to inflammatory gene expression in severe asthma, and thus could be used as a pharmacological target or a biomarker for glucocorticoid-resistant disease. So although my project was very specific, we can kind of use this as a framework to interrogate other immunomodulatory genes that are resistant to or even enhanced by glucocorticoids to find new therapeutic options. So thank you so much to my supervisor, Dr. Robert Newton, and all the people in my lab. Also, if you want to join us in Canada, we are looking for a new postdoc that has a passion for bioinformatics, so please come join us. Thank you so much. And if we have time for maybe one question?
Are there questions from the audience? Thank you, Amanda. That was beautifully done. Maybe I'll just ask a question. as it relates to whether or not you've had a chance to look at function yet with the activation of the IKKE epsilon in the, especially like the air-liquid interface cultures, barrier function, or something along those lines, the implication, if you will, of activation of this arc of NF-kappa B signaling. Thank you.
Yes, exactly. So that's ultimately, yeah, what I want to look into. Unfortunately, it has been kind of difficult because we don't actually know in airway epithelial cells what activates it. So I've done some like preliminary Western blotting, and I've found that it is phosphorylated in some conditions, but it doesn't seem to be reproducible yet. So yeah, we have to kind of throw a bunch of things onto the cells to actually figure out what conditions it's activated in order to find out what it does. But ultimately, once I do figure that out, we'd like to do a full sequencing where we knock out IKKE epsilon and figure out what it's actually doing in these cells.
Thank you.
Would it be possible to ask a quick question?
So, microphone one, please.
Hi, it's here, the one. Congratulations on the beautiful work. And we know that using these cell lineages is really a very important tool in order to investigate the mechanisms of responses. But I wonder, use the A549, whether we can extrapolate, let's say, the same mechanisms for epithelial cells from primary epithelial cells.
Yes, exactly. So we did find that at least that same induction did occur in the primary cells. So we are seeing the same regulation of IkB-KINASE epsilon, which we found in Bs2B cells as well. But some of the like transfection and cloning methods that I do would be tricky to do in primary cells, but ultimately it would be good to confirm those findings in primary cells.
Okay, thank you.
Thank you.
One more question at microphone 2, please.
Hi, beautiful talk, really beautiful. So well done for giving your first international talk on such a stage. I just have a question about the primary epithelium, because you're probably aware that you have steroids in their culture media. So hydrocortisone is always present in the differentiation media. So I was wondering if you'd done experiments where you'd removed that to see if it had your steroids had a greater effect or if it was different.
Yeah, that would be a really good idea, because I don't actually personally work with like the airway liquid interface, but I would like to be more hands-on in that process. So that would be a great idea to maybe get more of a reduction, because they're already going to be in the media. So that's a great point.
Sorry, thank you very much.
Thank you.
Amanda, you've done a wonderful job, and we're all very proud of you. Thank you very much.
Thank you so much.
Well, ladies and gentlemen, we move on to the next talk by my co-chair, Bruce Levy. Bruce is the Parker B. Francis Professor of Medicine at Harvard Medical School. That's a position that K. Frank Austin held, I think, sometime during his career. And he's also Chief of the Pulmonary and Critical Care Medicine Division at the Brigham and Women's Hospital. And we had a question earlier on about resolvins, and so here we have the expert who's going to tell us a bit about them. So thank you very much, Bruce.
Thank you, Professor Holgate, and thank you to the organizers. It's so wonderful to be here in Glasgow. Specific shout out to Francesca Levy-Schaefer for the invitation, and thank you for this Friday audience joining us for this exciting and interesting session. So my task is to tell you a little bit more about specialized pro-resolving mediators as agonists of the resolution of lung inflammation. And I know resolution has been a topic in several sessions over the course of the week. So I'll give a brief bit of background, but we'll jump straight into data from there. And although the title says lung inflammation, because of the focus today on asthma, I will focus in on the airway with the data that I share. I do have a few disclosures related to some equity positions, consulting work, and our grant funding. And before I dive in, I wanted, first of all, to thank Professor Holgate for recognizing Frank Austin, who just recently passed away. He's been an important mentor to myself personally, but to many of us in the field of asthma research and also bioactive lipid mediators. Professor Holgate pointed out already some of his incredible contributions, but in addition to Professor Holgate and myself, our next speaker also benefited from mentoring and support in career development from Professor Austin, so he's had a profound impact in the field. The field of bioactive lipid mediators has unfortunately also just tragically lost 2 additional members of our scientific community. Ken Hahn, who actually worked very early on monoheats and their influence on inflammation in cancer biology, way before the development of any immune-directed therapies for cancer, was way ahead of his time, and was a real leader in promoting careers in bioactive lipid mediator research. And then tragically, about a month ago, we lost Hilda Arnold-daughter, who, much too young, who died. One of the folks, relatively early stage career, although she just won a docent position at the Karolinska, working on SPMs as it relates to cardiovascular system. So I'll dedicate the talk to those individuals. So briefly, we've come very accustomed to understanding the pillars of inflammation: fever, erythema, edema, pain, and as Virchow said, loss of function if we don't intervene fast enough. Turns out that there are pillars of resolution as well, which summarized here by the R letter: removal of microbes, dead cells, and debris, restoration of vascular integrity and perfusion, regeneration of tissue, remission of fever, and relief of pain. And so resolution is now well appreciated to be an active process, not just the passive removal of pro-inflammatory factors. Biochemically, when one studies this with an unbiased approach using mass spectrometry, of course, in this ideal outcome of self-limited acute inflammation all the way to resolution and restoration of homeostasis, if you just focus in on the arachidonic acid metabolism, early during the upslope of inflammation, you see abundant formation of prostaglandins and leukotrienes, whereas later, the same parent fatty acid gets converted to structurally and functionally distinct metabolites that are known as lipoxins, concomitant with resolution. So if one takes resolving exudates in this theoretic space where neutrophils are decreasing and monocyte macrophage numbers are increasing, and if you sample exudates here, you see the emergence of products, bioactive lipid mediators, that were not present early in the inflammatory response. Lipoxins being one, and this broader array of essential fatty acid metabolism that has become known as the SPMs as a class, as a genus. But I'm showing the parent lipoxin, lipoxin A4 in its structure, and then E-series and D-series resolvins, the initial description of these members of a larger class of SPMs. And the DHA metabolome is broader than just resolvins, including protectins and merisins, and I'm showing you the parent molecules, protectin D1 and merisin 1 in this slide as well. Yesterday, Hongyong Pei from our group talked about one of the D-series resolvins as it relates to allergic lung inflammation, resolvin D2. So today, I'll be sharing you some specific vignettes related to lipoxin, resolvin, E1, protectin, and merisin. And in some cases, they're cystenyl conjugates. These bioactive mediators engage specific receptors, many of which, but not all of which, have been identified. Excuse me. These are expressed in a cell type specific manner, and they have some ligand specificity, but as you can see, some of these actually recognize more than one of these SPMs. And this leads to cell type specific actions based on their expression. While the entire signal transduction coupling is not well understood yet, there are some shared themes. To pick up on the last talk, for example, NF-kappa B is potently regulated by these SPM signaling in most cell types. There's elicitation of specific microRNA profiles, and in some cells, heme oxygenase 1 is actually induced. This slide just briefly depicts cellular mechanisms as it relates to the lung, and we're distinguishing anti-inflammation from pro-resolution. And so in red are anti-inflammatory actions of this class of molecule related to the granulocytes, as Dr. Pei talked about yesterday, sensory neurons as well. And then in purple here are innate lymphoid cells, where these molecules have a bivalent action, inhibiting pro-inflammatory cytokine production, but actually inducing amphoregulin release. And then in blue are pro-resolving actions as it relates to macrophages, NK cells, and regulatory T cells. And I'm going to share with you some of the data related to those in this talk. So what I'd like to do is start with this nice review for those of you interested in the area by Catherine Godson, who gave a talk earlier this week and is here at the meeting, on the balancing the effect of leukotrienes in asthma. Of course, Frank Austin was very involved in the identification and pharmacologic description of the cystinyl leukotrienes, and there have been drugs developed, as Professor Holgate mentioned, in this pathway. They've been helpful, but not uniformly so. And what we found in this set of work that Catherine summarized for the clinical implications of basic research series in the New England Journal of Medicine was that there's more than just cystinyl leukotrienes as it relates to this larger group of cystinyl-containing lipid mediators. As in this set of work, we identified in human lung meresin conjugates that are glutathione addicts, as well as protectin glutathione addicts, PCTRs. So the biosynthetic pathway is shown here. So instead of leukotriene A4, there's an epoxy meresin intermediate that leukotriene C4 synthase can actually attach glutathione to. It turns out that this molecule, another glutathione S-transferase, mu type 4, is more efficient with the DHA-derived biosynthetic precursor than LTC4 synthase, but both enzymes can do it, generating MCTR1, meresin conjugate of tissue regeneration 1, and then similar to the cystinyl leukotriene conversion of LTC4 to D4 to E4, you see conversion of the molecule to MCTR2 by gamma-glutamyl transferase, and then also from MCTR2 to 3 by dipeptidases. Turns out these molecules were active in terms of human lung contractile events and all in a preclinical model. And I'll just show you a couple data points from this. So Raja Abdulnoor in the lab led this work. And here we've taken precision-cut human lung sections that preserve their contractile activity and exposed them to a concentration response of leukotriene D4, which here with vehicle, you can see we get about a 50 to 60% narrowing of that airway in the presence of the escalating doses of, or concentrations of leukotriene D4. And then here you can see when we have MCTR1 or 2 or 3 present, the resulting change in the LTD4 stimulated airway contraction, with MCTR3 being quite potent at inhibiting the leukotriene D4 mediated contraction. So in the lung, there are counterbalancing, if you will, cystinyl meresins for the cystinyl leukotrienes in the human lung. We looked at this in this preclinical model of allergen sensitization and challenge in the mouse, where we allow the animal to get fully inflamed, in this case with chicken ovalbumin, and then at that point begin to administer MCTRs, in this case a cocktail, all 3 at once. Here's the airway hyperresponsiveness to methacholine dose response in naive animals, no sensitization and challenge. Here's the fully engaged animals with allergen sensitization and challenge. And you can see that the respiratory resistance by the flex event shows that the MCTRs, when given in vivo after development of the AHR, accelerate its resolution, again with MCTR3 being the most active molecule in this case. So they're promoting the resolution of AHR to methacholine, and not shown in this data set, but I refer you to the publication. They also promoted the resolution of type 2 lung inflammation and edema, the barrier permeability changes. So we've looked at several different mouse models of allergen-induced lung inflammation, and in work with Ollie Howarth, here pictured, when he was in the lab, we did an exhaustive survey of as we stop giving allergen and the eosinophilia in the lungs are decreasing over the course of a week, what is increasing? What is helping to clear these cells? And it turned out, to our surprise, that CD3 negative NKp46 positive cells, natural killer cells, were doing this. They were increasing in lung and mediastinal lymph node. And these cells appeared to be important for the clearance of eosinophils and antigen-specific T cells. When you depleted these cells, it impacted adversely the ability of resolvin E1 to promote resolution. So they seem to be important cellular mediators of SPMs in this context. We translated this early work to human severe asthma with Cindy Barnick initially, and we obtained peripheral blood and natural killer cells and looked first to see if they expressed receptors for SPMs, and they expressed receptors for both lipoxin A4, this ALX FPR2 receptor, as well as resolvin E1, the CMKLR1 receptor. And here you can see in healthy, mild asthmatic, and severe asthmatic, you could see that the ALX receptor in particular was distinctly increased in the setting of severe asthma, so we wanted to look at that in more detail. So we exposed either NK cells or autologous granulocytes to lipoxin, and what we found is that it enhanced NK cell cytotoxicity for the granulocyte, inducing apoptosis for eosinophils and neutrophils, and that this was receptor activated because the WRW4 peptide, which is a competitive antagonist at ALX, blunted the lipoxin's ability to do this. Lipoxin exposure to the granulocytes themselves did not induce apoptosis to any significant degree. So the apoptotic cell then becomes a substrate for clearance from an inflamed tissue by efferocytosis, macrophage-mediated clearance. And so this is a pro-resolving cellular mechanism for NK cell-mediated granulocyte apoptosis, engaged in this case by lipoxin A4 as a representative SPM. Because innate lymphoid cells are another innate lymphocyte that are present in circulating blood, we went and looked for these at the same time we were studying the NK cells, and we found these lineage negative cells in peripheral blood that also express the prostaglandin D2 receptor CRTH2, which were defined as ILC2s. This was the first description of their presence in peripheral blood, and we saw them healthy, mild asthma, and severe asthmatics. It turns out these cells also express ALX FPR2 as well as the resolvin E1 receptor. So we studied the functional implications of that with SPMs in isolated ILC2s from healthy donors. And when we activate the ILC2s, these lineage negative CD127 expressing cells, just non-specifically with PMA and A23187, a divalent cation ionophore, we see a burst of interleukin-13 production, a type 2 cytokine from the ILC2. If you, by comparison, when you look at NK cells, either dim or bright NK cells, we did not see the IL-13 production. So we looked at receptor-mediated agonists for these cells. They need IL-2 to survive in culture. So we looked at the epithelial alarmins, IL-25 and IL-33, and there was a significant induction of IL-13 from the ILC2s. Prostaglandin D2, remember these cells express CRTH2, by itself, of course, a mast cell product, induced IL-13. And when you put the epithelial alarmins and the mast cell agonist, receptor-mediated agonists together, you see synergistic increases in IL-13 release from the ILC2s. In black here, we saw regulation of this process by lipoxin A4, and not shown on this slide. It was receptor-mediated as well. So the ILC2s are responding to relevant cytokine stimulation in the airway in asthma, and this is also inhibitable by lipoxin A4. Well, one thing that was different about severe asthma than non-severe asthma or health was the ability of the NK cells to actually kill the granulocytes as a clearance mechanism. And you can see this is impaired in NK cells that we obtained from severe asthmatics. So we wanted to look at that in more detail. And Melody Duval in the lab led this series of experiments, where as part of the NIH's Severe Asthma Research Program, we were able to obtain bronchoalveolar lavage fluid cells to study the cellular makeup and immunophenotyping, as well as some function. And here what we found is that if you look at the relative amounts of Th1 cells to natural killer cells in healthies, non-severe asthmatics or severe asthmatics, you can see that they're increased. That ratio is increased in severe asthma. Here's Th17s, and here's neutrophils to NK cells. And in red there, in all cases, the severe asthmatic had much more of these effectors, these leukocyte effectors of non-T2 inflammation, these CD4 T cells and neutrophils, relative to their natural killer cells, suggesting that this NK cell clearance mechanism was not working properly in severe asthma. I want to take a moment. Is there a mouse? I may not be able to do this. Shoot. Well, there's a movie that I need a mouse to activate, but it doesn't seem, oh, there it goes, good. So the microtubule organizing center is shown here, where the granules, the cytotoxic granules of the natural killer cell get aligned, and perforin then gets put forward to the immune synapse to poke holes in the target cell membrane so that the package of granzymes and granulysins can be delivered to that target cell for killing, for cytotoxicity, so it's effective. And so we hypothesized that corticosteroids actually could adversely impact this process, and that the SPMs, through these ligand-mediated mechanisms that I've been describing, could actually help this process. And that, sure enough, that is what we found in this recent publication in Journal of Immunology with Melody, that dexamethasone impairs and lipoxin promotes NK cell lytic granulopolarization to immune synapse. This is showing confocal microscopy with perforin labeled here in green. And you can see when a natural killer cell engages a target cell, the perforin granules are close to that immune synapse. In cells that are exposed to dexamethasone, you can see that this does not happen effectively. The perforin granules are displaced from the immune synapse and never find their way there, whereas with lipoxin, you see this enhancement, if you will, of perforin granules at the immune synapse. We went on to show through a series of experiments that this is related to differential actions of the glucocorticoids on limb kinases, limb kinase 1 and 2. And these are important regulators of F-actin, this fibrillar actin, which is important for stabilization of that immune synapse. Lipoxins induce this process, glucocorticoids impair it. Well, there's been a lot said about virus already in this session, and we know viruses are important in the setting of asthma and asthma control. And I've just been showing you data that natural killer cells, which are important in virus host defense, are not working properly in asthma. So we wanted to study this in more detail. And so we used a clinical strain of RSV that in mice elicits a type 2 response, RSV line 19. And with this inoculum, we see at day 3 some early inflammatory changes, especially in the airway, that peaks at day 6, and then by day 12 is really coming back down towards baseline. If you look at plaque-forming units, you can see that they peak at day 6, and again, they're not gone, but they come back down by day 12. So there's an endogenous antiviral system. IL-13 mirrors the plaque-forming unit trajectory. And then if you look at ILC2s in the lungs of these animals, on the left, these IL-13 positive lineage negative cells, and on the right, the Th2, CD4 cells, you can see that the ILC2, IL-13 producing cells go up before the CD4s, but they have a pretty similar time course. So I mentioned that we identified in human lung the protectin conjugates of tissue regeneration earlier with that Morizen story. And so we're interested in protectin D1 and PCTR1 in this set of experiments. And that's because PD1 has been identified, protectin D1 has been identified in exhaled breath condensates and is a potent regulator in experimental allergic lung inflammation. Other groups have shown that protectin D1 can inhibit influenza viral replication, and as mentioned, the PCTRs are detectable in human lung. This work was led by Kate Walker in the lab, and this will focus in on airway epithelial cells. Here's the model is the same one that we used, except now beginning at day 3 after infection. we introduce PCTR1, protectin D1, or a vehicle. And we look at day 6. And both the RSV N gene expression as well as L gene expression is decreased by PCTR1 and PD1. This is the level of inflammation we see at 10x and 40x in vehicle-exposed animals. And you can see there's robust inflammation, especially around the airway, shown here. But with PCTR1 and protectin D1 at the level of the lung tissue, we see much less in the way of inflammation at day 6. Eosinophils are decreased, neutrophils are decreased, actually NK cells are decreased, and NKG2D positive NK cells are decreased, which are important for killing virally infected cells, suggesting that this is amplifying other antiviral mechanisms. Cathelicidin from naive in black to the vehicle-exposed animals in gray goes down. This antimicrobial peptide in the airway goes down in expression with infection, and it's partially restored with PCTR1 and PD1. Interferon gamma, the type 1 interferon, goes up with infection, and it's brought back down with PD1 and PCTR1. And interferon lambda, the type 3 interferon, is decreased by infection and partially increased in vivo by PD1 and PCTR1. In human bronchial airway epithelial cells, Calu-3 cells, what we see is if we in vitro expose the cells to virus, we get just a little bit of interferon lambda expressed in these cells. But when we expose to PCTR1 and PD1 in these virally infected airway epithelial cells, we see a burst of expression of interferon lambda. These are the first endogenous molecules identified to date that actually induce interferon lambda, this type 1 interferon. So in the setting of RSV infection and the associated inflammation, after infection, if you add in the airway PCTR1 and PD1, we see less RSV, we see less inflammation, and an enhancement of interferon lambda. And this is just a reminder that our first line of defense is this type 3 interferon from the airway epithelium. And it is protective by inducing the interferon stimulated genes without activating the subsequent inflammatory response. We became very accustomed to the type 1 interferons that we saw so much of with COVID, because not only was this inducing the interferon stimulated genes, but also activating what became a very problematic host inflammatory response. So there's some advantage to actually preceding that with the type 3 interferon activation, and this has not been lost on the pharmacology of PEG interferon lambda as it's been studied now in outpatients with COVID-19. This first study that was published in the Lancet Respiratory Medicine, in the blue line, you can see with higher levels of viral load to the right, the proportion of patients that became negative for SARS-CoV-2 RNA was higher in those who were receiving PEG interferon lambda. And then, excuse me, in the New England Journal of Medicine a few months ago, we saw the results of this phase 3 clinical trial with early treatment of the pegylated interferon lambda for COVID-19. And in orange here, in this summary of the trial results, in blue is the placebo, there were lower numbers of hospitalizations and ED visits within the 28 days of infection. And this was true whether people were vaccinated or not. And there was lower COVID-19 related hospitalization and death, and there was no significant difference in adverse events. So this very promising study also emphasizes the importance of these type 3 interferons in viral infection. And you heard the impressive work related to interferon beta in the last talk. I want to end with a vignette that focuses in on regulatory T cells, and this was led by Nandini Krishnamurthy in the lab, using the same RSV model. Day 3 now, we're going to give Marizin 1 rather than the protectins. And again, we see that with Marizin, there's less inflammation when we look at day 6 relative to vehicle. When we look at ILC2s on the left, CD4 TH2s on the right, producing IL-13, These are potently regulated by Merizin-1. RSV-L gene is also decreased with Merizin-1, and interestingly, we see this increase in amphoregulin by Merizin-1, which we didn't see with the protectins. And this amphoregulin is a mucosal protective cytokine for the airway. When we looked for expression of the Merizin-1 receptor, which had recently been identified as LGR6, in addition to macrophages, where we would expect the receptor to be expressed in abundance, when we looked at lymphocytes, we found this was highly expressed on regulatory T cells, more so than other CD4s, and that's shown by the flow plot here, and more so than CD8s. So there were low levels of other CD4s and CD8s expressing the receptor, but also mock infection to day 6 RSV to day 10 RSV, this was highly regulated by the RSV infection in regulatory T cells. You can see the pattern of expression in macrophages and other leukocytes here at the bottom. So this caught our eye, and we did a reductive experiment where we took cells, naive Tregs from healthy mice, innate lymphoid cells and Tregs from RSV-infected mice at day 6. And in those animals who received Merizin-1 in vivo, we took their Tregs out as well. And when you take the RSV ILC2s and activate them in culture, you see profound expression and release of IL-13. If you take naive Tregs that have preserved immunosuppressive function, that decreases the IL-13 release in culture by these ILC2s. RSV-infected Tregs lose this immunosuppressive function, and so the ILCs are able to increase their IL-13 production. Those animals who receive Merizin-1, their Tregs have regained this suppressive function for IL-13. And this is a TGF-beta transmitted activation and regulation by Tregs on ILCs, as it's inhibitable, even in naive Tregs, using a SMAD3 inhibitor, CIS3. So this suggests that the immunosuppressive function of the Tregs can be recaptured by exposure to Merizin-1. Well, we were able to obtain LGR6 knockout animals, a receptor knockout. And just when you infect these animals with the RSV, on the right there you see that they have more inflammation in the large and small airways in the knockout animals. If you look at IL-13 levels in lung tissue, in brown here's the knockout, black's the wild type. You can see that's increased. Here's ILC2s and TH2s in brown relative to gray. This is the wild type, the knockout, increased and increased just on its own. Here you see that the wild type has the Merizin-1 responsiveness in both ILC2s and in TH2s, but you lose that Merizin responsiveness in the wild type, the knockout animals. We also took human precision-cut lung sections. As mentioned, this RSV strain is a clinical strain of RSV. And then we take the human precision-cut lung slice from a pediatric source and expose to either media or the virus, and the virus in the presence of vehicle or MAR1. And you can see that with vehicle, you see this increase in IL-13, and you get loss of some of the tissue architecture in this viral infection. This is decreased substantially by Merizin-1, and the architecture is preserved in the Merizin-1 exposed lung. And this is also in part related to a burst with Merizin-1 of amphoregulin. So what I've tried to do with these series of vignettes is highlight how SPMs can be agonists for resolution of lung inflammation, and like an orchestra, I've highlighted some cell-specific targeting of the different mediators as we're coming to understand how they can propel resolution. So the MCTRs, I've shown you data on airway smooth muscle, the PCTR1 and PD1 on the airway epithelium, lipoxins, Resolvin E1 on NK cells and ILCs, and then Merizin-1 most recently on regulatory T cells. So we're trying to put these pieces together to understand how best to characterize an immunophenotype diseases of acute and chronic inflammation. So to summarize, we presented evidence that SPMs display bronchoprotective, anti-inflammatory, pro-resolving bioactions. They engage specific receptors to transduce cell type specific responses. And they target airway structural cells as well as innate and adaptive leukocytes. So in conclusion, these SPMs and the cysteinyl containing SPMs can regulate prophlogistic cellular responses in the airways, properties that potentially could be leveraged to target specific pro-resolution pathways of inflammation in airway disease. And of course, I did almost none of this work. I have worked with some wonderful, wonderful people in the lab. I've tried to highlight them along the way, and I've bolded their names here as well. We have important collaborations with Charles Sirhan, who leads the Center for Experimental Therapeutics and Reperfusion Injury at the Brigham, and these colleagues in his lab. Nikos Patasis, a chemist at USC, has provided us with authentic material to use in early forms of our experiments. And then we have great collaborations with the Asthma Research Center, led by Elliot Israel at the Brigham. I want to thank the members of the Severe Asthma Research Program and, of course, our patients who participate in that, and our funding sources from the NIH for the work I've shared with you today. Thank you so much for your attention.
Thank you very much indeed, Professor Levy, for that wonderful overview, which really kind of takes us down a very exciting new set of pathways, which I'm sure we're going to hear a lot more about. We've got time for one or two questions, if anybody has anything they would like to put to Professor Levy. Yes, sir, in microphone one.
Yes, Marco Aurelio Martins, Oswaldo Cruz Foundation, Rio de Janeiro, Brazil. And my question concerns your second model, RSV in mice. And I wonder if you have got any data and a sort of dual insult of the virus plus allergen-induced asthma. If you think that could modify somehow the mechanism implicated and the difficult to treat mechanism concerning sensibility to corticosteroids, for example.
It's a wonderful question, and we've modeled co-infection or subsequent infection, virus and bacteria, and studied SPMs in that context. But for the specific question that you're asking, what we're trying to do currently in the Severe Asthma Research Program in human subjects is to try to study that with both airway biospecimens as well as peripheral biospecimens and look at that intersection and what is being activated or what is being failed to be activated of these endogenous homeostatic mechanisms in that context. So we're trying to look in human subjects to address your question. It's a really important one. We could model it, and perhaps that'll be part of our future work, but we'd like to go straight to humans if possible for that.
Thank you very much.
Yes, thank you. Yes, Francesca.
Thank you, Bruce, for your overwhelming and nice lecture. I'm thinking, and I'm asking you actually to comment if you want to treat asthma, human asthma, severe, non-severe, which of the lipid mediators that you show have activities you would choose, because you choose many in your studies, and I wonder, we have a battery and we should choose, don't we?
Well, it's actually a complicated answer. So I think part of what I showed really would suggest that choosing any one, based on our understanding in kind of homeostatic self-limited responses that are more acute than chronic, would be challenging. Either a cocktail or some other way to more durably activate a pivotal homeostatic mechanism will probably be required. There is currently 3 fairly interesting and active pro-resolving therapeutics strategies that are moving forward. One relates to a lipoxin stable analog that's been looking, the investigators are looking at mouthwash actually for gingivitis and for periodontitis, and with excellent phase 1 and early phase 2 data. There's another group that took monocytes and radiated them so that they became apoptotic and created a gamish that they don't really fully understand. But that apoptotic cell mix is showing early, excellent preclinical data. And then there's a company in France that has generated an agonist antibody against the resolvin E1 receptor, which is moving quite rapidly through development, and it looks really exciting in targeting IBD. It may be that a mimetic that is stable or one of these agonist antibody approaches may ultimately be the way forward. But right now we're going to immunophenotype human severe asthma and try to understand if there are any pivotal hubs or regulatory checkpoints amongst these pathways that we could more selectively target. But you could see the multitude of the actions of these SPMs make it difficult to say there's just one, and also it's like picking just one of your children. Very hard to answer. Yeah.
Lovely.
What a comparison.
Thank you for the question. Not an easy question to answer.
Yeah. Thank you very much, and that was a very good way of finishing that excellent talk. Thank you, Bruce, for that. So, ladies and gentlemen, we come to the final talk now, and this is, I think, a very appropriate time to just say one or two things about Francesca Levi-Schaefer, because I've known her for many, many years. You heard that she has worked with K. Frank Austin 2 or 3 times during her career. She's been backwards and forwards to Boston, which has been always very nice for everybody. But I think the important thing really here is to say that she's the Myrna Kay Clinical Chair of Immunopharmacology in the University of Jerusalem. And I said that K. Frank Austin was the father of immunopharmacology. You were the mother of immunopharmacology. And of course, you set up the immunopharmacology group here at IUFAR a couple of years back, and you currently chair that, which is wonderful, and you will be the president next year or next time. So congratulations, and we look forward to hearing your talk. Thank you very much. The inhibitory receptors on mast cells and eosinophils as new targets for anti-asthma therapy.
Thank you very, very much for your flattering introduction. I wish to be the mother not only of my children, but also of immunopharmacology. It's too much for me. I also would like, first of all, to thank the audience remaining for the last session and the last lecture of the session, and the speakers of this session that has been carried out under the immunopharmacology section at the time, now committee of IUFAR. And yes, I would like to dedicate my presentation to the late Dr. Frank Austin, who made me more and more interested in muscle during my postdoc at Harvard, but also to Professor Barry Kay that Stephen mentioned, with whom I spent my first sabbatical as a Brompton. And to both of them, they show my way, muscles, and later on eosinophils. And because of that, today I'm talking about the 2 cells. I'll try to be quick. I also to catch a plane, so it is egotistically. So, also, yes. So, another thanks is to the previous speakers that explained so well about all the players in allergic diseases, possible players, cell players, mediators, and so on. So I can go quite quickly with some of the slides. What is important in this one, I think, is our observation that in asthma, but also, of course, in other allergic diseases, but also in several other diseases with different etiopathogenesis. I just to mention one in lupus, that is absolutely an autoimmune disease, in psoriasis, In IBD, mast are usually associated with eosinophils, so you find them both in the inflamed tissues. My research goal has long been to determine new immunopharmacological targets, principally for the treatment of allergy, nevertheless, and especially focusing on mast cell and eosinophils as key effector cells of these conditions. Any cells is important, of course, in an orchestra, but somehow the conductor is the most important guy. And therefore, we focus our studies on mast cell eosinophil, and on what I will show is the allergic effector units, is their crosstalk. And my lecture will be a short journey to explain what we found and why we arrived to the conclusion that it is a good idea to harness a new receptors that we found expressed on these cells, an activatory one actually to be decreased, and an activating, and the two inhibitors ones to be activated. I don't have to show you any more of these slides because Paul Ehrlich has been already shown. But in this slide you can see a picture that I took when I was a postdoc in Frank Austin, a picture of a metachromatically stained mast cells from nasal polyps, and it looked just like a heart. And since then, I decided my scientific love is mast cells. Also, in this slide, I show a few old papers of our group showing that mast cells are long-lived cells, that after activation, they regenerate their mediators and also their capability to be reactivated both by IgE and also non-IgE dependent mechanisms. So that led to the hypothesis that they might be important not only for the initiation of allergic inflammation, but also for for the late phase and for its chronicity, when they meet mostly the eosinophils. So this is an oversimplified view, of course, of allergic inflammation, where I point out that the center of the universe for me is the mast cell and the eosinophils, but it's also important to show that the cells that I'm talking today about have been shown by various researchers and us to have a crosstalk also with the smooth muscle cells, with the fibroblast, with the epithelial cells, with endothelial cells, so with structural cells, besides having crosstalk, of course, with other immune cells that are found or penetrating the tissue. But again, the most important, at least in Th2 asthma, persisting cells, and again, effector cells, is eosinophils. I also put in this picture bacteria, viruses, and fungi. We heard about viruses earlier on, and I would like to remind you that also bacteria, and especially S. aureus, is a very major infecting microbiota in allergic diseases, and the fungi as well. So we have to remember all this complexity, but try to go down to simpler pictures. And it's not because I am today here with Bruce Levy, but because I really believe that we have a new tool in resolving allergic inflammation, and I spent 2 sabbaticals visiting professor with Bruce, who taught me all about what I'm going to show today, or at least a big part of what I'm showing today is that we did in collaboration. So luckily, I don't have to go through what are the drugs nowadays for asthma. When I talk about asthma, of course, I'm talking mostly about moderate severe asthmatics, because the mild asthmatics, I think, are taking care of the older strategies that are always used and successfully used. But I would like to remind you that we have a newer strategy. I mean, anti-IG antibodies is already not so new. 15 years ago already, as omalizumab has been put on the market. Anti-IL-5, anti-IL-5 receptor alpha, targeting the eosinophils. You have anti-IL-4 receptor and anti-IL-13. You have many new drugs that were added to the treatment of either patients resistant to steroids or more severe. However, still we don't have a great drug for everybody, because even in the high IgE patients, anti-IgEs that was meant to stop asthma, it doesn't. Indicating, I think, that some other mechanism, and I will show you these two, others than IgE are present in the asthmatic lung and can activate muscle, showing that eosinophil, blocking eosinophil is not all the story. Not because it's always better, as Paul Ehrlich taught us, to stop at the beginning, to avoid the continuation of the disease. So if you want to stop the beginning, you have to stop the muscles. But because, again, eosinophils are not responsible for all the symptoms or clinical symptoms of asthma. For example, for the hypertrophy or the bronchial muscle, for the hypersecretion of mucus, and so forth and so on. However, in line with the previous lectures, I think that epithelium has been forgotten for a very long time, and now we have a new drug, at least for TSLP, one of the alarmins. You talk about IL-33, and I'm very looking forward to more data on this relatively new drug. Besides that, there are all a sort of other kind of strategies, and to either to stop the muscle or the eosinophils. One that has been entered in phase 2 is anti-Siglec-8 antibodies, however, not for asthma. It was unsuccessful for asthma, unsuccessful for atopic dermatitis, and now it's in trials for mastocytosis and chronic idiopathic urticaria. Of course, there are KIT inhibitors, and of course, because it's very modern, there are JAK inhibitors, JAK1, JAK2, JAK3. So what I'm trying to show you here is that we are not at the end of the road, and new kind of treatments such as the SPMs are very, very welcome. So, okay, so the allergic effector unit. What we noticed a few years ago was a physical proximity of muscles and eosinophil in many allergic tissues. And then we have postulated the hypothesis that muscle and eosinophil could interact. They are close by, they stay for a long time in the tissues, and they communicate possibly what they have to say to each other. When I first wrote the first grant on this topic, I say, well, in the 60s, I think, eosinophils were claimed to contain histaminases, and then to be good guys, to be there just to stop histamine activity secreted by the muscle. Muscles, well, produce, I don't know, heparin, let's say, so maybe they act as anti-inflammatory through heparin via the eosinophils. So it was not so clear that their crosstalk to be either pro-inflammatory or pro-resolutory. But from our first observations that show us, for example, the muscle-specific mediators such as tryptase can activate eosinophils via PAR2 to produce pro-inflammatory cytokines that, on the other hand, eosinophil contains preformed stem cell factor that is a most important survival and activator, pro-activating factor of the muscle. By the fact that eosinophil can activate Ig desensitized muscle via MBP, major basic protein of the eosinophil, show us that this interaction, at least with the mediators that we found and the receptor we defined on each other's cells, is a pro-inflammatory one. However, when we went to the physical interaction that we saw it take places, and it is the most precise interactions that you have in immunology, and not only, we, beside finding again an activating couple, CD48 and CD244 or 2B4, we could also define the presence of inhibitory receptors such as Siglec-7, CD300A, and the corresponding receptor, leaving us with a question mark. Maybe these two cells, besides being a friend in the inflammation, may be friends also in its resolution. Please tell me how much time I have, and I will go maybe faster towards the resolution of the lecture and resolution of inflammation. So the first observation on the physical crosstalk that we defined goes mostly through CD48, a GPI molecule present on the muscles, and 2B4, a SLAM-related protein, also the CD2 subfamily present on eosinophil, show us that muscle and eosinophil are forming units very fast in a durable way, and this ligand receptor couples is number one responsible for this physical contact. Importantly, the physical contact showed to induce eosinophil survival. Eosinophils are not so much long-lived as muscles. They need their growth factor, IL-5, GNCSF, IL-3. And indeed, we found that also in the physical-induced cell survival, GM-CSL produced by muscle was very important, but it was not all the story. And in fact, this eosinophil muscle increase eosinophil survival is not stopped by dexamethasone, also linking to the presentation of IKKε. The physical induced contact induces cell activation, and you will see this in next slide, as just shown for increase in tryptase release. So if we have isolated cord blood muscles and we activate with Ig-mediated mechanism, we have a 40% roughly of tryptase release, and if we add in co-culture and have contact eosinophil from peripheral blood, you can see how the release of tryptase is increased. And it is partially decreased by anti-2B4 and also other receptors such as ICAM1 and Nectin-2. Same happens with eosinophils. These are eosinophil culture alone, even not activated. The co-culture with muscle activates them further to release EPO. However, we could see that blocking antibodies that would block the physical contact didn't block this increased activation. So we think that for the muscle increased activation is important the physical contact between muscle and eosinophil and the receptor we show, beside others. For eosinophil activation due to muscle in vitro activation to degranulate, soluble mediators are more important, and we just show in collaboration with a group of Marcus Mauer that indeed if you take supernatants of IgE-activated human skin muscle, you can stimulate CD69 expression on eosinophil, and this stimulation is absolutely blocked by anti-GM-CSF, also showing, I think, to the market of anti-IL-5 for eosinophils or anti-IL-5 receptor, that there is a more activating survival factor for eosinophil. And some maybe of the failures seen because of treatment of eosinophilia in Th2 might be due to the additional activity of survival factors for eosinophils produced by muscle and by other also tissue cells other than interleukin-5. So we show how important CD48, and I will quickly go to convince that why we didn't choose 2B4. I don't go into the papers we publish on that, but the first idea, the CD48, to block CD48 would be a better target than to block 2B4, came by study, seminal study by Bad Zimmerman and Mark Rothenberg. They show the CD48 is one of the 291 mouse asthma signature genes, and not 2B4. And then we saw very importantly that CD48 as expressed by human, muscle, and eosinophils is a ligand for S. aureus and for its exotoxins. And therefore, I said, okay, let's look at CD48. Indeed, when Ariel Muniz, at the time a PhD in my lab, did a model of alum in mice of asthma, and he delivered anti-CD48, anti-CD2, or anti-2B4 antibody, blocking antibody, only the anti-CD48 and not the other antibodies could block both the sign of inflammation in the bronchi and the sign of mucus production, but also, as you see, only IL-5 could be reduced, IL-13, and the total bulk eosinophil, but only by the treatment of blocking CD48. In a study we did in collaboration with Peter Blooding and later on with Stephen Holgate, we could see the couples of muscle and eosinophils or allergic effector unit in lung of asthmatic patients of various severity, and to colocalize CD48 both on eosinophil as stained by MBP and on muscle as stained by tryptase. More recently, we saw the importance of CD48, not only actually for Th2 asthma, in asthmatic patients having chronic sinusitis and nasal polyps, very much increase in the group of non-allergic asthma with rhinosinusitis in comparison to non-allergic allergic asthma or only allergic rhinitis. again, CD48, in this case, on eosinophil. So in the big studies that we could carry out thanks to Stephen Holgate, we ask whether the membrane form of CD48, because being a GPI, it exists both as a membrane form and a soluble one, could have differential expression on cells of the blood. And we saw that even if it's not necessary a marker of Th2 asthma, it was a significant increase in mild and moderate patients, not on neutrophil, not on basophil, not on B cell, T cell, and NK, while surprisingly on severe asthma, B cells, T cell, NK, and monocytes, we had an increase of CD48. We were curious if we could design CD48 as a marker of asthma, but it's not convenient to use a membrane marker. Therefore, we look for the soluble form of the mediators that we thought it will correlate with the cellular expression. And with the UK cohort, we have the big surprise that in severe asthma, let's start, in mild and moderate asthma, you have an increase of soluble CD48, but in severe, these levels of soluble CD48 goes down, as well as you see if we divide the inhaled steroids treated patients, and even more if it is a glucocorticosteroid oral treatment. Therefore, one would say, haha, you give steroids and of course you decrease the level of the soluble, but that was not true, since what we found it is that soluble CD48 is released by a mechanism, phospholipase D and C, that is not controlled by the steroids. So it seemed to be an inherent property of severe asthmatic cells. Moreover, so what we say, why should CD48 be high in mild, in moderate, and so low in severe? And we found that the soluble CD48 is a decoy receptor for 2B4, as shown, for example, here on eosinophil. If you stimulate eosinophil with an activating 2B4 antibody and you put soluble CD48, you have less EPO release and less L-alate release and less VAV phosphorylation. So there is a physiological function of soluble CD48 that is not produced in asthma and is not produced enough by the mild asthmatic. I try to finish because of the time. Quickly, my lecture, I will concentrate now on one of the inhibitor receptors we found expressed both on muscle and eosinophils, and try to link it at the end with resolution of inflammation. Now, CD300A, it belongs to the immunoglobulin superfamily of inhibitor receptor. It has for I-team, it was previously described by Moretta group of NK cells, neutrophil, a subset of T and B lymphocyte, and we, in collaboration also with other groups, found expressed on muscle, eosinophil, and basophil. Now, the ligands, the no ligands, are phosphatidylserine and phosphatidylethanolamine that are expressed by apoptotic cells and or by activated cells. So it renders for me this receptor very important in inflammation at all. I don't go into this slide just for the one who might have forgot. Inhibitory receptors are phosphorylated by the activating receptor and become active, recruit phosphatases such as HCP1, SHIP1, or 2, and they dephosphorylate very quickly upstream activating receptors. So are really ideal drug to my view, because they are very upstream. Therefore, during the years, we decided to target CD300A specifically on muscle and on eosinophils, since, as I told you, they are also expressed on NK cells. And also in a different way, it is done in cancer therapy. In cancer therapy, you want to block the blockade of inhibitory receptors to try to activate NK and T killer cells, and we want to target with agonistic antibodies the inhibitory receptor in order to inhibit muscle and eosinophil. So at the time, there was no molecular engineering. We synthesized bispecific against CD300A or IgE or CCR3 or c-KIT, and in elegant studies done by Ido Bachelet and Ariel Muniz in my lab, we could show the mechanism of course of these antibodies in vitro, in vivo, and we could see, relevant to these lectures, that there was abrogation of allergic reaction and asthma models by these specific antibodies. Recently, we found that CD300A is increased on peripheral blood eosinophil of asthmatic patients, moderate, and this shows that our body, together with the production of pro-resolving lipid mediators, is also upregulating inhibitory receptor on the fetal cell of allergy, of asthma, to try to downregulate. Okay. And now we are at the newest generation of bispecific antibodies that we obtained by the cross-mab technique. They are engineered antibody, and we try to go on with our studies. Okay. Now, I can go fast. I don't need to explain to you anymore what is resolution of inflammation. This is a part that we develop together with Bruce Levy. And I just would like to tell you that our hypothesis was not only that you have a deficiency of course, of these pro-resolving lipid mediators in asthma, but that maybe muscles are pleiotropic. Maybe muscle at a given point don't succeed enough to resolve the inflammation they started with, and there's a crosstalk between CD300A and lipid mediators. Therefore, in a model of a peritonitis model of alum, we saw the CD300A knockout mice indeed have impaired resolution, as shown by persisting high numbers on inflammatory cell and mostly by the eosinophil, and have an interesting production of lipoxin A4 and expression of the receptor on the cells. We found also together in the lab of Bruce that exogenous SPMs can decrease human and murine muscle activation, indicating indeed that human muscle have a receptor for at least here, for resolving D1 and lipoxin A4 that have a shared receptor. Two slides, last but not least, also we showed together that lipoxin B4 promoted the resolution of airway inflammation in the upper and lower airways of mice, comparable, if not better, to the one you have with dexamethasone. So the last slide, I promise, before the last slide, I would like to show the very exciting new findings that we just published in pharmacological research, that mast cells, it doesn't matter if are cord blood derived, if foreskin derived, if nasal polyp derives, as soon as they are activated by Ig mechanism, and only by Ig, by the way. We tried, if you remember, Bruce, with papain, with compound 4080, but only Ig, so the same stimuli that causes allergic inflammation starting, they can produce quite significant quantities of resolving D1. We target resolving D1 because there is an ELISA kit. Now we are doing a lipidomics also with Chasiran. And this production continues up to 72 hours. We continue the experiment up to 72 hours, perhaps it stays longer. And we notice that the 15-lipoxygenase, the enzyme responsible for the production of resolving D1 in presence of DHA, also stay stable. So I open a new avenue. I hope that in 2 years, next IUFAR meeting, we will show what happens in the allergic effector unit, if indeed the muscle has the power to downregulate allergic inflammation by CD300A expression and by resolving D1 and other lipid mediators. It is a commentary that my student at the time, Per Giorgio Puzzovio, and I wrote, interpreting data showing JAK in practice and trying to explain via our allergic effector unit, you have or you don't have a good activity of omazumab and mepolizumab in asthmatic patients, type 2 and muscle activation syndrome. So I suggest if you could to read, is very short. So I don't go again to the conclusion. I think I show you enough about the importance of CD48, of the allergic effector unit, and of inhibitor receptor. But what I think is very important, and again, I learned this from Bruce, that SPMs are released in a time-dependent fashion. So if we want to treat asthma, we don't have perhaps at the very beginning to give SPMs, to activate CD300A. We have to think also about the timing of the treatment and try to lead the inflammation not only to stop the inflammation, because these lipid mediators, as it was mentioned, are not anti-inflammatory only, at least. They are pro-resolutory. They help us to go back to homeostasis. Now, being thanks to my group, these are the last components, to you for being here. And you see, if you come and visit my lab, that I hope you will, in a beautiful place such as Jerusalem, you will be given as a souvenir a thermos showing us the allergic effector unit and with what we believe, or I believe, it must have been love. Thank you very much.
Well, thank you very much indeed, Francesca. I think you brought this symposium beautifully together at the end, really, by showing these cell-cell interactions, which are sort of critical, really, in a complex disease like asthma. So maybe just one or two questions, if there's anybody got anything they want to raise. There's a gentleman over here coming up to microphone 3, and then microphone 1.
Thanks very much, Francesca. I was really interested in your mast cell eosinophil interactions, your physical interactions, and I guess from the Southampton lab, one of the defining aspects of severe asthma was mast cells right in amongst the airway smooth muscle cells. Now, I don't remember there being eosinophils in there, so I was just wondering is there sort of selective interaction in the lung between eosinophils and mast cells? And if there's no eosinophils in the airway smooth muscle with the mast cells, does that suggest perhaps different roles for the mast cell in that particular setting?
Yes, thank you for asking me the question. I'm very familiar with the Southampton studies, and I've been asked this question already. I think I have to demonstrate as a scientist that in the mouse muscles, cells, in the muscle cells, mast cells, I don't pronounce correctly mast and muscle, yeah, you are right, are there in the absence of the eosinophil. We might think that eosinophils still influence them by soluble mediator from below or upper parts, and yes, and we think that this interaction, because we saw, and Peter Brading elegantly shows that, is located somewhere else in the bronchi, but contribute to the ongoing of the disease. But we have to study the muscle in different locations. It is more complicated than that because it has been proven by Swedish researchers that muscle in different lung locations have different phenotype. Some are totally mucosal type muscle because you have much more mucus in the lung, but you have some also parenchymal connective tissue muscles. Anyway, we found the resolving D1 is produced by both equally, and CD300A also it is expressed by both equally, both a kind of muscle. So, thank you.
Thank you very much.
Thank you very much, Professor Olivier Schaffer. Very nice data. I was fascinated by this idea of mast cells being important also in the resolution phase of the inflammatory response. I wonder if you have got any data concerning the this resolving in mast cells, if they are there preformed or neo-synthesized, and if this mechanism of IgE-mediated release of resolvins would be sensitive to mast cell stabilizer agents.
Yes, thank you for your question. Yes, we are investigating now if you have also preformed source of resolving D1, but by doing a full lipidomics of the muscles, in which you study also the cell associated, not only the release. But usually, you teach me, these mediators are newly formed, newly synthesized. So we don't think, until proven otherwise, they are already ready to go. But they are quickly produced. Our experiment were activation of muscles 30 minutes just because of convenience of time, but also earlier times they are produced and released. And muscle stabilization is also a very important point. Might reduce, you would say, this release, but we found for the moment at the level of messenger RNA for the enzyme that by activating CD300A and therefore stabilizing muscle doesn't reduce the 15-lipoxygenase expression and hopefully activity. So you can have a differential treatment of these cells.
Well,
Thank you.
Thank you very much. You've given us tremendous and provocative data to consider that all of us can take with us to lunch, and thereafter that mast cells may be involved not only in the initiation of the allergic response, but also potentially its resolution. And with that, I'd like to close the session. Thank you all for your attendance, and thank you, Francesca, for putting together such a wonderful session and meeting. Might I invite the speakers to come up to the stage very quickly for a quick photograph, and then everyone enjoy lunch, and the rest of the meeting, I understand Patrick Valance will be giving a lecture in another hour or so. I'm sure that'll be very interesting. Thank you, and safe travels home, everyone.
Thank you.